Hello, this is Mr. McLean. In this video, we're going to go through the early steps of the cell reproduction chapter. We're going to be talking about, um, the different stages of interphase, as well as the stages of the cell cycle that include mitosis and cytokinesis. So, before we get into that, want to make sure we all focus on the idea that when we talk about cell division or cell growth and reproduction, we're talking about one of the key characteristics of all living things. And regardless of what type of life you're describing, they are going to definitely have some method and mechanism for how they're going to make new cells. So, one of the more primitive approaches are what the prokaryotes use, and it's called binary fission. And if you take a look at the picture here, binary fission is essentially going to make a clone of whatever you start with. So this is what we do whenever we are looking at bacteria under most instances where it's able to grow very rapidly and increase its number very quickly. So there's not a lot of genetic diversity that's going to take place here because you're getting clones, but it definitely allows a population to build very quickly. So this is something that we see in the prokaryotes. So that would be the bacteria. Now, on this slide, we're taking a look at two methods of reproduction that are going to take place with our eukaryotes. So the one on the left is showing us mitosis. And whenever we think about mitosis, we're thinking about reproduction for our body cells. So whenever you have some type of injury or growth that's taking place, that's going to be for mitosis to generate the new cells that are necessary. The one on the right is showing meiosis, and meiosis is going to be what is used for generation of the sex cells. So whenever an individual is getting ready to reproduce, to generate those gametes, they have to use meiosis. So we'll get into the details of that later on. So one of the important things to look at for any type of cell and any of the steps of cell reproduction is you have to look at the genetic information. So DNA is universal with all living organisms. You all have to have some type of genetic information. So whenever a cell is not in the process of going through replication, the DNA is coiled up in what we call chromatin. So if you look at the picture in the bottom left, you can see inside the cell, they have this chromatin. It almost looks like spaghetti inside of there. And associated with that chromatin are going to be some special proteins called histones. And what we're going to notice is that whenever a cell gets ready to replicate, the DNA is going to coil up around those histones and take on more of a shape of that X-like structure that you see on the bottom right, which is going to be a chromosome. All right, so here's a couple more images. You have a clear picture of a chromosome on the far left. And the diagram in the middle, if you start on the right side, you can see from DNA, and the DNA is wrapping around these proteins called histones. Those are little red things. And then eventually it coils up and it forms chromatin. And then if it's really condensed and coiled up, it will form those chromosomes. So it's super important to understand the structure of a chromosome. And if we look at this picture before we get into any of the text, the top picture, the left side of it shows you what is typically represented as a chromosome, just like the one at the bottom of that particular screen. And if you look at that compared to the picture where you just see like a single chromosome, the difference is the duplicated chromosome actually has two arms that have been or two chromatids, as we should really call them, that have actually been copied already there. So whenever we bring together the DNA and the histone, it's going to form what's called a chromatid. And when we take a look at a fully duplicated chromosome, it's going to be made up of two chromatids that are going to be clones of each other. So when we talk about DNA replication, we're talking about being able to copy the DNA exactly as it was and generating it into a new molecule that's going to be exactly the same as what we started with there. So in this particular image, with the duplicated chromosome, the two chromatids that make up that chromosome are called sister chromatids, and sister chromatids are identical to one another. Okay, and then that little pink thing is like the connection point. That's called the centromere. So the centromere is where we join the two chromatids together. So the format of our DNA is going to be influenced very heavily by what's going on in regards to the cell cycle. Now, another thing that I do want to introduce is the idea of homologous chromosomes. So if we go to the right side of the screen, we're going to see that we have um, two homologous chromosomes that together make up what we call a pair of homologous chromosomes. So we have the purple and the green. And a good way to think about that is, just for simplicity's sake, let's say in this picture, green chromosomes are the ones that you get from your father and purple are what you get from your mother. So for a human, if they have chromosome set number one, they got one chromosome in all their cells that came from their mother, and they got one chromosome number one in all their cells that came from their father. So when you put those together, they make up a pair of homologous chromosomes. The purple and the green have the same genes on them, but they are not identical. And obviously, your parents are not identical. So the coding between the purple one and the green one is going to be slightly different, which gives us our variations. But if there's a particular trait that's covered on the purple one, it's covered on the green one as well. So whenever we look at species, one of the things that is kind of a defining number or factor of a species is how many chromosomes they have. And if the organism is reproducing sexually, it's going to have chromosomes that are in pairs. So that would be why we call them homologous. So the offspring between two individuals are going to have chromosomes in pairs. So for the sake of a human, we have 46 total chromosomes in our body cells. That's 23 pairs, 23 that came from mother, 23 that came from your father, that give us that grand total. Okay. Now, here's a couple more images of that. So you can see here, they're saying maternal means mom, paternal is dad. So there's the pair for number 11 and pair for number 17. And once again, they cover the same traits, but genetically, they are not going to have the exact same coding because they came from different people. Now, if we go in here, this is an important term that you want to make sure you understand as we go through the process of mitosis. It's the idea of diploid. So if you look at the picture, diploid is represented as 2n. N is the number of sets, and 2 is saying that there's 2 for each one of those sets. So in our body cells, which are known as somatic cells, those are going to be diploid. On the right side of the diagram, we have haploid, and haploid is 1n. And that would mean that there's only going to be one chromosome for each set. So if you go through there and we have, 3 sets of chromosomes, that would be what the grand total of chromosomes that you have. So that would be an example of a sex cell, otherwise known as a gamete. Now, when we get into sex cells and sex chromosomes, and we start to look at some of these different things, this picture right here is an example of what's called a karyotype. And a karyotype is a picture of all of an individual's chromosomes. And if you take a look at it, this particular person has 22 chromosomes in the pink area, and those are called autosomes. They actually have 22 sets. So that would be 44 total chromosomes. So there's pairs, homologous pairs for each of these. The bottom right corner, you see it's in a light blue color. Those are the sex chromosomes. So in this particular instance, this would be the karyotype for a male because there's an X and a Y, and the females would have X and an X for that. Okay. So the sex chromosomes are the ones that are associated with sex determination. So most humans are going to have exactly the same number of chromosomes. They're going to have 46. And one of the things that can be done is to verify that a picture of an unborn child's chromosomes can be actually analyzed and looked at. So in this particular picture, you'll notice that every chromosome set has 2, which means that there is a grand total of 46 chromosomes. If we had a child who had Down syndrome, they would have what's called trisomy number 21. And if you look at number 21, there would be 3 chromosomes there. So that's not the case in this particular instance. But when we talk about changes in chromosome number, usually we're talking about serious mutations. So as we move on, we start into the cell cycle. And the cell cycle is how we go through and we take a look at some of these different phases of the cell cycle. We're going to see that in this list, we have G1, S, and G2. They are all collectively known as interphase, right? So that's a big part of what we're looking at in this section of the chapter. Interphase is when the cell is growing and making necessary adjustments, but it's not dividing yet, okay? So this is the non-division stages. Here's another way of looking at it. pretty much everything except for that mitosis cytokinesis is non-replicating. So interphase makes up the vast majority of the time. And you can kind of start there. G1 is first, followed by S, G2, and then mitosis and cytokinesis. So it's the longest phase without a doubt. And it starts off with first growth, which is technically G1 stands for first gap, but you can just kind of think of it as first growth. You're going to have a lot of growth of the cell. You're going to have a lot of organelles increasing. You're going to have a lot of the enzymes. The things that the cell needs are going to get larger in number. Now, I have this statement at the bottom about G0. G0, we're going to come back to, but that's whenever a cell doesn't progress to cell division, but it also moves past G1. So that's a special scenario that we'll look at here in a minute. S, you want to think of S as synthesis, and the synthesis is making DNA. That's DNA replication. So that's really, really important. That's when, if it's going to be determined that the cell is going to divide, you're going to have to make sure you have double the amount of DNA. So S phase is really important. And then after S, you have G2, where you have additional growth, and you get some of the things that we're going to see in the second video, where we talk about centrioles and microtubules, and our cell's just going to get bigger, and it's going to be prepared for cell division. So those are all things that will happen. Now, when I mentioned G0, what I wanted to focus on is some cells, they kind of get that G1 and then they don't progress further. They actually kind of come out of the cycle, and we call that area of that progression of the cell existence as G0. So two major types of cells that don't replicate after initially being formed, that would be anything dealing with nerves and your muscle. So that makes it super critical that we be very cautious about spinal cord and brain trauma issues, because that is potentially going to be an issue. All right. Now, controlling the cell cycle, we know that it's really critical that we have some type of control mechanism, checkpoints, if you would, to make sure that we're not going to kind of speed past certain things. So there's three main checkpoints. There's one at the end of G1, and that's kind of like the all or nothing one, to be honest. If it makes it through G1, it's most likely going to make it through the other two. If it gets rejected at G1, that's when it bounces it out to G0. So you can kind of think of it as little red lights. So you start here and you make it up to that first red light. If you make it through, you're going into S phase. If for some reason that cell is unable to go into S phase, it steps out into G0. Now, most of our cells spend some time, and maybe even a lot of time, in G0. Think about your skin, for example. You're not constantly making new skin, but you replicate skin very frequently. So it can kind of step out of the cycle, and then it can be called back in as needed. And that's obviously going to be an important stage there. All right. So with that, we're going to stop this video. And in our next video, we're going to get into a discussion about the different stages of mitosis. So hopefully that was helpful on getting information about chromosomes and the initial stages of the cell cycle going there.